Sweet chestnut blight is a serious fungal disease of sweet chestnut trees caused by Cryphonectria parasitica. The primary host is sweet chestnut, which includes the species Castanea dentata, Castanea sativa, and Castanea mollissima. It can, however, also affect various oak species and has been recorded on Eucalyptus viminalis. Cryphonectria parasitica is native to Eastern Asia, where it causes little damage. It was accidentally introduced into North America in 1904. Since then it has spread over the native range of American chestnuts and killed about 4 billion trees. Cryphonectria parasitica is currently in Europe and it has been very damaging on European sweet chestnut. As sweet chestnuts are a commercial crop, it is important that the disease is controlled. Currently in Britain we do not produce very many sweet chestnuts, but as the climate changes it may become a more lucrative crop. There have been outbreaks of sweet chestnut blight in Britain. Cryphonectria parasitica was identified in November 2011 as the cause of dieback in European sweet chestnut plants that had been planted in Warwickshire, central England. Approximately 300 trees had been planted out for nut production in 2007, and an infection became apparent in about half of the plants. A second outbreak was later found on a smaller site in East Sussex, which had about 30 trees, and had also been planted out for nut production. Both sites had sourced their trees from the same place. In fact, on the slide you can see the plant passport label that accompanied one of the trees. Plant passports are supposed to ensure that the trees are healthy. However, in this case they were not, and had been accompanied by some unwanted passengers. It really illustrates that even regulated pests can slip through the net. All the diseased trees were located and had been eradicated, and as far as we know, there are no current outbreaks in the UK. Up to 2013, 10 sites had been confirmed, and all of these were in nurseries. There were never any outbreaks in the wider environment. Cryphonectria parasitica attacks the bark of European sweet chestnut trees. It is a wound pathogen and tends to enter through fissures and wounds. Once it is in, though, it starts to kill the bark and form a lesion. Stems or branches can be girdled by this lesion and die. The dead bark becomes visible as a sunken canker, as illustrated in the photograph on this slide. Above the canker, the leaves wilt, turn brown and die, although they usually remain attached. Below the canker, the tree often reacts by trying to produce epicormic growth. In this slide we have some photographs of trees with typical symptoms. You can see in the image on the left there is a small area of brown leaves. This is the leaf collapse above a lesion. In the photograph on the right, you can see a roughened area of bark, which is the lesion, and epicormics sprouting from the trunk below. So if you were to come across a tree which had been affected by Cryphonectria parasitica, this is what you might see on the bark. The image on the left-hand side shows what you would see with the naked eye. These orangey or brown structures emerging between the cracks in the bark. If you were to take out a hand lens and look closer, you would see what is illustrated on the right-hand side. There are orange or yellow to reddish-brown pustules emerging through the lenticels in the bark. The lenticels are the tree's gas exchange pores. The fungal fruiting bodies produce long tendrils of spores, particularly in humid conditions. In this slide you can see the orange patches of fruiting bodies on an infected branch. These photographs were taken by our plant health officers. In the image on the right you can see that the bark has been removed, and beneath the bark there is dead brown tissue. Another characteristic symptom that you might come across is the formation of pale brown mycelial fans in the inner bark. If you were to peel back the bark beneath which you had observed the orange fruiting bodies, you may well come across them. They are typically creamy white in colour and spread out in a fan shape. So what are the biosecurity measures for sweet chestnut blight? Well, it is important to follow full high-risk biosecurity measures. You should not remove any wood or tree materials or debris from a site where it has been confirmed, as spreading occurs via infected plants, wood or bark. The long-distance spread of Cryphonectria parasitica occurs through international trade, with infected plants, wood or bark acting as the pathways. Once the bark has been colonized, the mycelium of the pathogen can survive in the tissue for up to 10 months, even if it has been air-dried. There is also a small risk of transmission by fruit or seeds. Locally, the spores of Cryphonectria parasitica are spread in wind and rain, and that is why it is important not to sample in wet conditions, as this might aid the spread of chestnut blight locally. Chestnut blight can also be spread by other agents including insects and birds. Entry into suitable tissue for infection might be aided by wounds produced by insect vectors. It has been reported that Cryphonectria parasitica can exist as a saprotroph, i.e. it lives and feeds on dead organic matter, on broadleaf trees beyond its parasitic host range. This can allow it to persist even when infected chestnut trees have been removed. Chestnut blight is a wound pathogen, and so it can be introduced to new hosts during sampling if tools are not cleaned and sterilised. Cryphonectria parasitica is treated as a European Union quarantine organism, which means that movements of planting material must be free from it, with plant passports issued to provide an assurance of compliance with requirements and to ensure the traceability of material. A number of European countries, including the United Kingdom, are also protected zones for Cryphonectria parasitica, as listed in the EU's Plant Health Directive. Movements into these zones require special compliance with additional requirements, accompanied by specific plant passports eligible for the zones in question. In the case of Cryphonectria parasitica, the movement of round timber and isolated bark into countries with protected zone status is not permitted unless the bark or timber has been passported and is accompanied by an official statement to show that it has been kiln-dried or that the bark has been fumigated. New requirements for statutory notification of imports of Castanea, that's sweet chestnut, as well as Platanus, plane trees, Quercus, oak trees, and Fraxinus, ash trees, came into effect on the 17th of January 2013.